Okay, so welcome to this next video in which we are discussing complexins and neurotransmitter release. Okay, right, so far we've got to the stage where calcium has come in and 3 calcium ions are bound to the C2A domain of synaptotagmin and 2 calcium ions are bound to the C2B domain of synaptotagmin. Now, what is synaptotagmin going to do? Well, since this is a video on complexins, we'll present what it does to the complexins first. Basically, there is good evidence that synaptotagmin binds to complexins, these complexin proteins here that are bound to the snare proteins, and causes them to change conformation, okay, so that they now go from being this sort of clamp protein which was inhibiting the core snare complexes from fusing the two vesicles together, the two membranes together, to having a pro-fusion effect. So it's basically going to change the function of complexin from being anti-fusion to being pro-fusion. Okay, so this is important to understand. Complexin was once thought to be just this protein that was a clamp protein that had this inhibitory effect and stopped the core snare complexes from fusing the two membranes together before the action potential has arisen. Now what is emerging is that it must have a pro-fusion effect, and we're going to discuss this more in upcoming videos, because when you knock them out, when you knock complexin 1 and complexin 2 out, you get major problems, you get huge problems with exocytosing neurotransmitter. And in fact, we believe by knockout studies that if you remove these complexin 1 and complexin 2 proteins, neurotransmission, exocytosis of neurotransmitter goes down by 60 to 70%. You lose 60 to 70% of exocytosis. That is a major effect. And in fact, loss of function of complexin 2 is thought to be very much so involved in Huntington's disease and giving the phenotype, the disease phenotype of Huntington's disease. And we'll discuss that more in upcoming videos. Okay, so we know basically from knockout studies that these proteins have to have some sort of pro-fusion effect, basically. So our initial theories that this was merely a clamp protein went out the window when we did these knockout studies. Okay, so what we now think is that when synaptotagmin becomes activated by calcium binding to it, it binds to the complexin, changes complexin's function from being a clamp protein to being a facilitatory protein that's going to help the snare complexes move these two membranes closer together. Okay, so that's one of synaptotagmin's functions. Now we'll just have a revision of the other stuff that we've seen that synaptotagmin does. And I want to stress that all of these things are coming together to all contribute to the fusion event. It's not just one pathway that causes fusion. There are multiple pathways all contributing to trying to fuse these two membranes together. Synaptotagmin interacts with complexin 1/2. That promotes fusion. In addition, it's now going to do its other interactions. So for instance, we also know that synaptotagmin binds to syntaxin 1. So shall I make a list of these? It activates the complexin 1/2. Okay? We also know that it binds to syntaxin 1. We saw evidence of that in yonks ago. So synaptotagmin binds to syntaxin 1, this blue snare protein, and we can also understand why that might promote fusion. And then most importantly of all, potentially, well, actually both of these are important, but another important thing it does is it binds to lipids that are in this phospholipid bilayer and causes basically an invagination of the plasma membrane towards the membrane of the docked synaptic vesicle, and that bringing together of those two membranes is going to promote fusion between the two. So let's have a look at this binding to lipids in more detail. So basically, the C2A domain of synaptotagmin 1/2 is going to bind to a normal component of the phospholipid layer of the plasma membrane known as phosphatidylserine, and the C2B component is going to bind to a normal component of the phospholipid bilayer known as PIP2, or phosphatidylinositol 4,5 bisphosphate. So let me discuss with you the structure of those two molecules. Okay, right. So if this is the phospholipid bilayer, then the normal component of the phospholipid bilayer is normal phospholipids. So let me show you the structure of a normal phospholipid. So here is the cartoon structure of a normal phospholipid. So these these vertical lines here, these represent the hydrophobic tails of the phospholipid. Okay, so these are the hydrophobic tails, and they come about from the long-chain carboxylic acids, or fatty acids, which have been esterified to the first and the second hydroxyl groups of this glycerol molecule, which forms the backbone of the phospholipid. So here in green, this is the glycerol molecule. Okay, so in green, this is glycerol. The proper name for glycerol, or the chemist's name for glycerol, is propane 1,2,3-triol. Okay, 1,2,3-triol. And the nice thing about the chemist's name, even though it's a mouthful, is that it tells you exactly what glycerol is. It tells you that it's a 3-carbon molecule where you have hydroxyl groups or alcohol groups of every single carbon. Okay, right. So basically what you have is 2 long-chain carboxylic acids esterified to the first and the second hydroxyl groups of the glycerol molecules. These are fatty acids or long-chain carboxylic acids. They're the same thing. Right. Now, of the third hydroxyl group of the glycerol molecule, which I've drawn in orange here, this is a phosphate group. Okay, and it's linked to that hydroxyl group by a phosphoester or a phosphate ester bond. Okay, now this whole molecule is a normal phospholipid. So this is a phospholipid. Now there is an old name for a phospholipid, which you might still hear if you're a chemist, if you're a biochemist, not if you're a chemist. If you're a biochemist, you might occasionally hear this name used, but to be honest, phospholipid has replaced it. And the only reason it's useful to know this old name is because these other molecules, these modified phospholipids, are named via this old name rather than this new name. Okay, so the old name for a phospholipid is a phosphatidate molecule. So this whole structure here, the glycerol with the 2 fatty acids in the phosphate group, that will be referred to as a phosphatidate molecule. Okay, right. So now let's discuss what these 2 molecules that the 2C2 domains of synaptotagmin bind to are. So one of them is phosphatidylserine. Phosphatidylserine, and basically now that you know what phosphatidate is, you might be able to guess what phosphatidylserine is. So if we draw out another phosphatidate molecule, here are the long-chain carboxylic acids which are esterified to the first and second hydroxyl groups, and here is the phosphate group of the phospholipid esterified to the third hydroxyl group, and basically what you do is you stick a serine amino acid off this phosphate group here. You form an ester link between, a phosphoester link between the hydroxyl group of the serine amino acid and the hydroxyl group that is free of this phosphate group. Okay, so you stick serine onto this phosphate group like so. So now what you've got basically, oh dear, I've coloured it in wrong, sorry about this. So the red and orange have switched around, so the red now denotes the phosphate group and the orange denotes the fatty acid. Sorry about that. So here's our phosphatidate molecule, and then we've just stuck serine off, so it's just phosphatidylserine. Phosphatidate off of a serine molecule. And basically, these molecules can sit in the phospholipid bilayer just like a normal phospholipid. All they have is a larger head, basically, so it's just a modified phospholipid. So that's one of the components of the phospholipid bilayer which synaptotagmin can bind to. Specifically, the C2A domain of synaptotagmin is going to bind to this when it binds calcium. Okay, now let me discuss the other components. And by the way, phosphatidylserine is often just abbreviated to PS. Now the other component that the synaptotagmin can bind to has a longer name. It's still phosphatidylinositol, 4,5-bisphosphate is the name of the other component that synaptotagmin can bind to. Okay, right, bisphosphate. Right, so let me pull this up. So basically, again, this is a modified phosphatidate molecule. So let me draw it here. Here are the long-chain carboxylic acids, which are esterified to the first and second hydroxyl groups of the glycerol molecule. Here's the phosphate head of our phosphatidate molecule. So let me colour it in. So this is the phosphate group. I'm wondering if we're going to have enough space on here now. Here are the long-chain carboxylic acids, which are esterified to our glycerol molecule, which is in green here. Okay, and then we are going to link off this an inositol molecule. So let me draw this here. So here is our inositol molecule. Now, inositol is a molecule you might never have heard of other than in this molecule. And inositol is a 6-membered carbon ring, where all of the bonds between the 6 carbons are single bonds, okay? And off all of those carbons, you have an alcohol group. So if I was to draw the skeletal structure of inositol, it would be like this. Here's the 6-membered carbon ring, and then you'd have alcohol groups of every single one of the carbons, like so. Okay, and off all of the carbons you would also have a hydrogen, basically, but you don't draw that in skeletal structures, and in fact it makes it look far more complicated if you draw the hydrogens. It makes it look messy. So the skeletal structure highlights how simple this molecule is and how beautifully rotationally symmetric it is. Right, so that's an inositol molecule. Now what you're going to do is take one of these hydroxyl groups and form a phosphate or phosphoester link with the free hydroxyl group of this phosphate group that's bound to the glycerol. Okay, that creates you phosphatidyl inositol. Now we want to convert phosphatidyl inositol into phosphatidyl inositol 4,5-bisphosphate. So we need to add phosphate groups onto this. Now we're adding them onto the 4th and the 5th carbon. So this is counted as the first carbon, this one up here. So we count round 2, 3, 4. So here's a 4th phosphate group. 5. So here's a phosphate group off the 5th carbon here. So that then is the structure of phosphatidyl inositol 4,5-bisphosphate. And this is often abbreviated to PIP2. P for phosphatidyl, I for inositol, P2 for bisphosphate, phosphate times 2. Okay, right. And if you stare at this molecule enough, what you will decide, basically, is that that name is not good. It should not be called phosphatidyl inositol 4,5-bisphosphate. If you were following convention for how to name compounds, you would not name that phosphatidyl inositol 4,5-bisphosphate. You would name it phosphatidyl inositol 3,4-bisphosphate. I do not know why they named it 4,5-bisphosphate. If anyone does know, please comment away. I would be delighted to hear it. Regardless, we're stuck with that name. And this is the molecule that Synaptotagmin's C2b domain binds to. So let's draw this happening. So if this is our Synaptotagmin molecule here, okay, so this is Synaptotagmin here, then basically this is the C2a domain, and remember 3 calcium ions bind to the C2a domain, and this is the C2b domain to which 2 calcium ions bind. Now when calcium has bound, what can happen is these C2a and C2b domains can bind to these phospholipids, which are normal components of the phospholipid bilayer. So let's draw them in here. So let's draw this phosphatidyl serine in here. Okay, so this is a phosphatidyl serine molecule. So this molecule here, just drawn a little smaller. Okay, so let me colour it in to make it more obvious. So here in blue is our serine. Here in red is our phosphate group. Okay, and here in orange are the hydrophobic tails there. And in green, you then have the glycerol molecule. Okay, so that's our phosphatidyl serine, or PS for short. Now it's the C2a domain that binds to phosphatidyl serine, okay? And the C2b domain then binds to the other component, the phosphatidyl inositol 4,5-bisphosphate. And I want to stress that this happens only when calcium has bound to the C2A and the C2B domains. It doesn't happen prior to that. Okay? So it happens once the synaptotagmin has been activated. So there's our inositol ring. Here are our phosphate groups off the 4th and the 5th carbon, then the phosphate group of the phosphatidate molecule, and then here are the long-chain carboxylic acids, which are esterified to this glycerol molecule here. Okay, so this is PIP2 here, phosphatidyl inositol 4,5-bisphosphate. And the C2B domain is going to bind to that once it's had calcium bind to it. So I should show the calcium binding here. 2 calciums are going to come here, 3 calciums are going to come here. Okay, and basically this interaction with the lipids of the plasma membrane, and I want to stress that these lipids here, they are only present in the plasma membrane, the membrane of the cell. They are not present in the synaptic vesicle membrane. If they were, it would cause major problems with the function of synaptotagmin, because basically synaptotagmin would try and latch onto its own membrane, the membrane of the vesicle that it's in, and that wouldn't help us at all with exocytosis. So, it's very important that they aren't in the synaptic vesicle membrane. So they bind to these phospholipids these modified phospholipids in the phospholipid bilayer of the plasma membrane, and what basically they cause is they cause the membrane to invaginate inwards. So the whole membrane, the whole plasma membrane sort of invaginates, is pulled towards the synaptotagmin. So if I draw these little synaptotagmins here, then they're going to pull the plasma membrane towards them by pulling on these PIP2 and phosphatidylserine molecules. Okay, and that's going to pull this plasma membrane towards the membrane of the synaptic vesicle, and that is going to promote fusion of the synaptic vesicle with the plasma membrane. So this interaction with the lipids is also really important for the fusion. But I want to stress, remember these knockout experiments that we're going to go into in more detail. You knock out complexin 1 and 2, and what happens is you get a 60 to 70% drop in neurotransmission. So this movement of the phospholipid bilayer is important, but the complexin interaction is also very important. So these are all adding together to produce fusion. All of them are important. Okay, so I want to stress that point again. There's not just one mechanism that leads to the fusion of the plasma membrane with the synaptic vesicle membrane. There's multiple different mechanisms all contributing. If you knock one out, you don't completely abolish exocytosis of neurotransmitter, but you reduce it. Okay, so that's the end of this video, where we're just discussing complexins and neurotransmitter release. In the next video, what we'll discuss is knockouts of complexin and how it leads to, how we believe it's very, very much so involved in certain neurological degenerative disorders such as Huntington's disease.